Today I will review the concept of antigen. Practically any molecule which is able to generate antibodies against itself, we call such molecules as antigens. Antigens can be defined as antibody generating molecules, and these molecules can exhibit two types of properties. First one is antigenicity and second one is immunogenicity. If this molecule X is able to bind with specific antibody which is generated against it, we call this property antigenicity. If this molecule X is able to bind with antibody, we call this molecule X as antigenic. And if similar molecule X is able to enter our body, it is able to generate specific T cell or B cell immune response, we call this property immunogenicity. And we call this molecule X immunogenic. So the difference between antigenicity and immunogenicity is simply if a molecule X is able to bind with antibodies against it, we call it as antigenic, and if this molecule X is also able to generate B cell and T cell responses, we call this molecule X immunogenic. In immunological sense, we can define antigen is any molecule that express both these properties. Any molecule that express both immunogenicity as well as antigenicity. All immunogens are antigens, but not all antigens are immunogens. Let's take an example of this molecule X. This molecule X enters our body and it is able to generate specific immune response. We can call this X as immunogenic. And when these B cells release specific antibodies against this molecule X, anti-X antibodies are able to bind to this molecule X. We can say this molecule X is able to bind with these antibodies, so it is antigenic as well. This molecule X is both immunogenic as well as antigenic. So practically all immunogens are antigens. But there are many proteins, many self proteins in our body which can bind with specific antibodies, but still they do not generate specific B cell and T cell response. So all the antigens cannot be considered as immunogens, but all the immunogens are definitely antigens. Let's come to the another concept, which is concept of epitopes. What are these epitopes and why are these epitopes considered as antigenic determinants? Let's come back to this diagram again. This is a molecule X. It can be made up of proteins or it can be made up of carbohydrates, lipids, anything. But this molecule has to have certain specific sequences which are being recognized by these antibodies. And those specific sequences are known as epitopes. If the nature of this antigen is proteinaceous, there has to be a sequence of amino acids which are being recognized by specific anti-X antibody. And how is this antibody recognizing these specific sequences which we call epitopes? The variable region of this antibody is forming specific sites which are complementarity determining sites or complementarity determining regions, CDRs. Now these CDRs are the specific sites on the antibodies which are recognizing the epitopes on these antigens. Antigens have epitopes and antibodies have paratopes. Those regions of CDRs which are binding with these epitopes are considered as paratopes. So, antigenicity of any molecule is determining by the specific sequences present on its surface, and we call those specific sequences epitopes. And these epitopes are being recognized by CDRs of antibodies or even some receptors of B and T cells. Epitopes are part of pathogen or antigen which gets recognized by the CDRs against which the immune response occurs, which means that this antibody has been generated against this molecule X. That is why this is recognizing this specific sequences, specific epitopes on this molecule X. And second concept we already defined, that regions on an antigen which are recognized by paratopes, and these paratopes are also the CDRs of the specific antibody which is made against this specific antigen. Now, the important concept is number of epitopes on a particular antigen is determining the valency of that antigen, and higher the valency of the antigen, more will be the immune response against that antigen. So in short, we can say that valency is directly proportional to immunogenicity. which means if we have 10 epitopes, there will be higher degree of immune response as compared to antigen with 2 or 3 epitopes, let's say. Now let's define these epitopes by their nature. As you can see in this diagram, this is, for example, a proteinaceous antigen, and this is a folded protein. So this folded protein may have specific epitope which is exposed on the surface, and these exposed surface epitopes are called by their respective name, exposed or surface epitopes. Now there may be certain epitopes which are not exposed on the surface, which are lying internally. We call them internal epitopes. And if we talk about the structure, specific structure, and if we consider this type of antigen is proteinaceous in nature, proteins exhibit different kind of structures. Protein may present in primary structure, protein may present in secondary or tertiary structure. If it is primary structure, we have linear chain of amino acid, and linear chain we have a sequence of amino acid involved. That sequence of amino acids we call sequential epitopes. As you can see in this diagram, the sequence of amino acids is involved, and we call it as linear or sequential epitopes. If the secondary or tertiary structure is involved, we are including certain hydrophobic interactions, we are including protein folding, and protein folding may bring together different amino acids. For example, amino acid present on this side may be closer to amino acids present right here in a folded structure of protein. And such structure which are not linear, which are not in sequence, we call them non-sequential or conformational epitopes, because there is conformational change in the protein. Protein has acquired tertiary structure, and different amino acids have come closer to form this specific epitope. Now let's come to the properties of antigen. What are the different properties exhibited by that antigen, and how are these properties related to immunogenicity? The first property we will talk about is molecular mass. And if the antigen is proteinaceous in nature, we need at minimum 6 to 8 amino acids to form a single epitope on that particular antigen. And if it is carbohydrate in nature, that we need 6 to 8 monomers of a particular carbohydrate to form an epitope in carbohydrate type antigen. Suppose we are given 4 different proteinaceous antigens with molecular masses: A 1 kilodalton, B of 10 kilodaltons, C of 50 kilodaltons, and D of 100 kilodaltons. And we are asked to arrange them in the order of immunogenicity. Which protein out of A, B, C, D will exhibit highest degree of immunogenicity? So the concept here is higher the molecular mass, higher the size, higher the complexity, and the molecular mass is highest in this D peptide. So this D will exhibit highest degree of immunogenicity. Whereas this A is only 1 kilodalton, is almost non-immunogenic in nature. So order of immunogenicity will be D is most immunogenic and A will be least immunogenic. The reason is practically simple. Greater the molecular mass means this protein might have highest number of amino acids, and more the amino acids, more epitopes they will form, because 6 to 8 amino acids will form only one epitope, and more the amino acids, more epitopes will be formed. And larger size also allows the protein to be folded into tertiary structures, which will exhibit more complexity, and more complexity lead to more immunogenicity. This leads us to the concept that as we increase size of the antigen, immunogenicity will proportionally increase. Now let's come to second property of antigens, which is complexity. Now what are the factors which are deriving the complexity of these antigens? Let's take an example of 4 different types of antigens. We have proteinaceous antigen, we have carbohydrate made antigen, we have lipid and nucleic acid antigens. The first thing we need to remember is nucleic acids are practically non-immunogenic. If it is a pure nucleic acid, it is non-immunogenic. And proteins exhibit highest degree of immunogenicity. Why? Because they are, firstly, they are made up of 20 different types of amino acids, and greater the number of amino acids, greater the independence to form the different secondary and tertiary structures, and its complexity will be increased several fold. That is the reason. Protein will be having highest degree of immunogenicity and nucleic acids will be having lowest degree of immunogenicity. Nucleic acids are practically what we say non-immunogenic in nature. So order of immunogenicity is protein highest, which is followed by carbohydrates. Lipids are least immunogenic. Nucleic acids are practically non-immunogenic in nature. Carbohydrates are moderately immunogenic because they have simpler structure as compared to proteins, and lipids have even simplest structure, so they are least or non-immunogenic on their own. But it has to be remembered, if these are complexed with each other, they may become immunogenic. For example, if we complex nucleic acids with proteins, it will become immunogenic due to the protein part of this nucleic acid. We call such nucleic acids nucleoproteins, which are complex with proteins. The more chemically complex a molecule is, the more potent and immunogen it will become. Now let's talk about the nature of polymers, because all these structures are polymer in some way. Amino acids polymerize to form proteins, monosaccharides polymerize to form complex carbohydrates, and so on. So what will be the nature of polymer which will exhibit the maximum degree of immunogenicity? So heteropolymers always exhibit highest degree of immunogenicity, and homopolymers are relatively less immunogenic as compared to heteropolymers. Now let's consider two different types of proteins. First protein is made up of, for example, 100 amino acids, and all those 100 amino acids are practically same, which means one amino acid is repeating several times to form a 100 amino acid protein. And second protein is also made up of 100 amino acids, but all 20 different types of amino acids are participating in second protein. Which will be more immunogenic? So it is pretty clear that the second protein, in which all 20 amino acids are participating, will be more complex in nature, and complexity will increase the immunogenicity. So a heteropolymer which is made up of different amino acids will be more immunogenic as compared to homopolymers which is made up of same repeating amino acids. This is the concept of complexity, and complexity is maximum in heteropolymers. Let's try out this question. The question is saying we are given with 4 different mice A, B, C and D. A is injected with Keholimpet hemocyanin KLH. B is injected with human immunoglobulin G. C is injected with LPS, which is lipopolysaccharide, and D is injected with dextran. We have to find out the order of serum IgG. We have to find out the immunogenicity with respect to the serum IgG. The question is asking about IgG, not IgM, because as we know, the first antibody which is generated is usually IgM and not IgG. To produce IgG, we have to class switch from IgM to IgG, and class switching is only occurring for proteinaceous antigens and highly complex antigens. So dextran is carbohydrate antigen, and LPS is lipopolysaccharide, which is a mixture of carbohydrate and lipid. So class switching will be least for these LPS as well as dextran. Now we have to find out which is more immunogenic in KLH or IgG. So KLH is Keholimpet hemocyanin, which is a large sized protein and it is heavily glycosylated, whereas this IgG is smaller in size and it is also glycosylated. These two are glycoproteins, but glycosylation is more in case of KLH. And due to its size and complexity, the immune response with respect to IgG will be highest in case of KLH. KLH is followed by human immunoglobulin G. Both of these are heteropolymers, so no doubt about that. Now, what will be the order in respect to LPS and dextran? LPS is heteropolymer and dextran is homopolymer. LPS lipopolysaccharide. It is having lipid as well as polysaccharide, so immune response will be more in case of LPS as compared to dextran, though both of these will have negligible class switching. So our final order will become KLH will be highest immunogenic with respect to IgG. It is followed by human IgG, then lipopolysaccharide, and least immunogenic will be dextran. Let's get to another question. We are again given with 4 mice A, B, C and D, which are introduced with different peptides. These mice are injected with different peptides. So mouse A is being injected with peptide which is having all D amino acids. For mouse B, we have more D amino acids as compared to L amino acids. For mouse C, we have more L amino acids as compared to D amino acids. Let's consider that the length of the peptide is same in all 4 mice, and D is injected with L amino acid only. Find out the order of immunogenicity. So the concept is our enzymes only function against these L amino acids, and immunogenicity will only occur against the L amino acids only. So to find out the answer to this question, we have to find out the order of L amino acids. L amino acids are purely 100% present in the case D, so D will be having the highest immunogenicity. It is followed by C because C is having more L as compared to D amino acids, and it is followed by this B. B is also having some amount of amino acids, and D amino acids, this which is the case of mouse A, will be having least immunogenicity. So the order of immunogenicity will be D greater than C, which is significantly greater than B and A is the least immunogenic in nature. Let's move on to very important concept. Consider an organism. We do not know the identity of that organism, but we extracted two different proteins from that organism. Let's name these proteins protein A and protein B. And we reintroduced both of these proteins back to this individual. So in a nutshell, we extracted two proteins from this individual, protein A and protein B, and we reintroduced both of the proteins back to this individual. According to our hypothesis, there should not be any immune response against protein A or protein B because these proteins were originally extracted from the individual itself. So these proteins are self to this individual, we can say. But was observed was that against protein A, there was no response, which is quite obvious. The reason is that the protein A is self to this individual, so this individual should not respond against protein A. But against protein B, we saw massive immune response. So what could be the reason that protein B is just like protein A, which is both of these proteins are self to this individual, but only against protein B we saw massive immune response. So the concept is there are specific sites in our human body, for example cornea, or some sites in the reproductive organs where there is no blood supply. And if there is no blood supply, the proteins of those areas which are lacking blood supply will not be having immune cells as well, and this leads to the concept of privileged sites. There is no blood supply, there is no reach around of these immune cells, there was no recognition at all, and these proteins in these sites like cornea will always be considered as non-self. This protein B has to be some protein of cornea where there was no blood supply, so this was considered as non-self protein. This organism do not recognize this protein as self protein and initiated a massive immune response against this protein. This is the concept of privileged sites, or we can say immune privileged sites. So these sites are never reached by the immune cells and they are considered foreign to the immune system. But normally immune cells never reach there, but we artificially extracted a protein from cornea and reintroduced back to this organism. That is why there is massive response against this protein B. This is the concept of self and non-self proteins with respect to the immune privileged sites. Next concept is that we extracted different proteins from fish and introduced it to amphibians, reptiles, birds and mammals, and we have to find the order of immunogenicity in these different organisms. The concept here is greater the phylogenetic distance, greater will be the immunogenicity. So phylogenetic distance is directly proportional to immunogenicity, so the order will be mammals will have maximum immune response against fish protein, it is followed by birds, followed by reptiles and amphibians. Now let's come to another concept, which is concept of conserved proteins. There are certain proteins which are conserved throughout the evolution process, like cytochrome C in humans is exactly same to cytochrome C in a fish. So if we extract the protein of a fish, cytochrome C of fish, and introduce it to humans, there will be no immune response. So this is exception of conserved proteins, that despite so many variations among different phylogenetic distances, there are certain proteins which are conserved. By conservation we mean that there is exactly same amino acid sequence between different organisms, so there will be no immune response. But we studied right now is the criteria for immunogenicity. First of all, we have to see the molecular mass and size of the protein or antigen. Larger the size, greater will be the immunogenicity. Then we move on to complexity. What is the nature of molecule? Proteins are the most immunogenic in nature, and these are followed by carbohydrates and lipids. Then we come to complexity of the polymer. The heteropolymers are more immunogenic as compared to homopolymers, and we also saw the nature of amino acids affecting the immunogenicity. L amino acids will always be more immunogenic as compared to D amino acids. We also talked about origin of these proteins, and evolutionary distance is increasing the immunogenicity. And we also talked about the exception of the conserved proteins. We also talked about the self and non-self concept with respect to the immune privileged sites as well. Let's talk about immunogenicity with respect to route of entry. This concept is important for the concept of vaccination. So there are mainly two routes of entry for the entry of pathogens. First one is intramuscular or intradermal, and second one is intravenous entry of pathogen. So let's talk about intramuscular or intradermal, and most vaccinations like BCG are administered through intramuscular or intradermal route. When the pathogen enters the tissue region, it is processed inside the lymph node, and there is the involvement of T cells, there is involvement of B cells eventually, and there is massive immune response. That is why vaccines are preferred intramuscularly, so that there will be massive immune response and the generation of memory B cells and memory T cells will take place. Second concept is for the intravenous, and vaccines are not administered intravenously, because in case of intravenous administration, most of the T cells will not respond. 80% of the T cells will not respond, and B cell response is also very limited, and this response will take place in the spleen. This is the main reason why vaccines are given or vaccines are administered intramuscularly and not intravenously, because intravenous administration can lead to very low immune cell response, very low memory, and the concept of tolerance as well. So there is intramuscular and intradermal administration, which is always way more immunogenic as compared to the intravenous administration of pathogen, or in case we are talking about vaccines. Let's talk about haptens. Haptens are the small organic molecules, or they may be proteins, they may be small organic molecules which are less than 1 kilodalton or less than 1000 daltons in molecular mass, and they are univalent in nature. They do not have epitopes, and if they have epitopes, they only have univalent nature. They have only one epitope. Haptens are lab synthesized. They do not occur naturally, and they are non-immunogenic in nature. By non-immunogenic, we mean these are the molecules which cannot elicit B cell and T cell response against themselves. So either there are no epitopes, or there is only single epitope, which makes them non-immunogenic, but they are antigenic. Antibodies can bind to these haptens. So they are antigenic, but they are non-immunogenic lab synthesized molecules which are having nature of either protein or small organic molecules. The molecular mass is less than 1000 daltons, and they are univalent in nature. Hapten is introduced in mouse, there is no immune response, but when these haptens are introduced along with another protein, for example, we have a specific carrier protein. This protein may be outside protein, or this protein may be the self protein of the mouse itself. And when hapten is introduced in complexion with such carrier proteins, there will be massive immune response against this whole complex. As you can see, with time, if we introduce hapten alone, there is no immune response at all, and if we introduce hapten along with the carrier protein, there is massive immune response, and it shows usual immune response curve. Examples of haptens: the most common example is 2,4-dinitrophenol. The carrier protein is usually bovine serum albumin BSA protein, and this complex leads to massive immune response as well. The concept is Haptens themselves are non-immunogenic, but they can be made to be immunogenic by introducing certain carrier proteins. So haptens must be covalently bind to large carrier proteins. Covalent binding is not always necessary. Covalent binding is only necessary in vivo systems, but in vitro systems non-covalent binding may also work as well. The hapten carrier conjugate or hapten carrier complex is large enough to elicit immune response, and there will be B cell response, T cell response, generation of antibodies. This is the basic concept. We introduced hapten by itself, there is no immune response. We introduced the self protein of mice into itself, there is no immune response to self protein. But when we conjugated the hapten with self protein, there is massive immune response, there is strong immune response. The carrier protein, which may be self protein or which may be non self protein, along with size, it introduces T cell epitopes so that T cells can recognize this protein, and without T cell help there will be weak or weaker B cell response as well. B cell recognize the hapten, T cell recognize the peptides derived from the carrier protein. This is the main concept. That is how these haptens are made immunogenic. There is generation of three different kind of antibodies. There is anti-hapten antibody, there is anti-carrier protein antibody if the carrier protein is external, and there is anti-hapten carrier conjugate antibodies which is against the hapten and carrier protein complex. There are three different kinds of antibodies which are produced, which are anti-hapten antibodies, anti-carrier protein antibodies, and anti-hapten carrier protein antibodies as well. And the most common example in real world is a drug is introduced into the body where it binds with a plasma protein. The plasma protein is the protein of the host itself, and an immune response is generated. So this specific generation of immune response is achieved by the introduction of haptens, which are specific for the carrier proteins which are present in the plasma, and the drug we call it as hapten in this case. Let's talk about super antigens. The goal of antigen or any pathogen is to multiply, survive, and evade the immune response against it. Super antigens are the antigens which behave different from the normal antigens, and they very well effectively evade host immune response. And to evade host immune response, they have achieved or they have developed additional capabilities like heat resistance, like enzymatic resistance. They are resistant to many proteases so that they cannot be broken down. If they cannot be broken down, they cannot be expressed through MHC presentation. So heat resistance, protease resistance, these are certain additional capacities or additional capabilities which are expressed by these super antigens. The origin of these super antigens are usually bacteria like Staphylococcus, and mechanism is to protect the pathogen, protect this bacteria from a massive immune response by the host. What are the properties of these super antigens? So these super antigens are very much heat resistant, they are protease resistant, which means they cannot be processed by the MHC system. Another very important property is they do not bind with the receptor itself. They bind away from the peptide binding sites of MHC2, and T cells will behave abruptly. So there is abrupt immune response against the antigens. The size of these molecules, super antigens, may lie from 22 to 28 kilodaltons. And as you can see, this is the T cell receptor, this is the MHC presenting some peptide, but super antigen is not broken down because of the protease resistance properties, but super antigens can bind away from this MHC presentation site. They are binding externally. There is abrupt behavior of these T cells. It binds to non-polymorphic domain of MHC2 on alpha polypeptide chain, but it will never bind to the complementarity determining regions, CDRs, which are actually recognizing some kind of antigen right here. The super antigen is evading the CDRs of these specific receptors. Abnormal immune response, but this immune response is not specific with this antigen, so that the bacteria or pathogen can evade this massive immune response. Multiple T cells can also bind to a single super antigen. This is also seen in many cases. There is massive cytokine release, there is release of human necrosis factor alpha, and there is release of interferon alpha, and there is massive inflammatory response. And if the person is somehow immunocompromised, this may lead to toxic shock syndrome, and the common symptoms are fever, redness, pain, and some systemic inflammation as well. So let's compare the conventional activation versus super antigen activation of T cells. Antigen is processed by the antigen presenting cells. They are processed and presented by this MSC2, and MSC2 are presenting it, which is recognized by the CDRs of T cell receptor, and there is certain co-stimulatory signals like CDR3B7 signaling system, which will lead to activation of these T cells. These T cells will also result in some effector cell responses, which may also involve the activation of B cells as well. So the scale of activation is it activates a tiny fraction of T cells that are specific to that one epitope, and outcome is a well targeted, well controlled and effective adaptive immune response, which involves T cells and B cells as well. But in case of super antigen, it is binding abnormally, it is binding away from the specific antigen presenting site, and this will lead to multiple T cells to accumulate and bind to this abnormally. This will lead to abrupt behavior of these T cells, and the final outcome is unregulated massive non-specific cytokine release will be there, like TNF alpha, interferon alpha, from the activated T cells. The patient is immunocompromised. This leads to massive cytokine storm and systemic inflammation. It may lead to some life-threatening circumstances like toxic shock syndrome as well.